Hello guys, how are you? Now we'll start a new chapter. Chapter 2, Transmission of the Genetic Information, Activity 1, about mitosis. Fertilization is the fusion of haploid gametes, ovum and sperm, each of one unequal 23 chromosome, in order to form the diploid zygote to unequal 46 chromosome. This zygote or initial cell or mother cells, which contains two unequal 46 chromosome, divides into two identical daughter cells, each of two unequal 46 chromosome. But how? The zygote divides many times, and the number of cells is multiplied. These divisions are called mitosis. Mitosis, cell division ensuring a conformed reproduction of the genetic information. That means which mitosis, these division are called mitosis, which transmit to the daughter cells the whole genetic information carried by the chromosome. An adult human contains an estimated 100 trillion cells, and yet we start life as a single cell. To grow, develop, and repair tissue damage, we rely on cell division. In eukaryotic cells, this process is accomplished by a series of well-orchestrated steps called mitosis. Every day, our bodies must produce millions of skin cells to replace those lost through normal activity. Each of these cells must have a complete complement of the genetic material. Before cell division, where the cell divides into two identical cells, the DNA needs to be replicated or duplicated so that each daughter cell receives an exact copy of DNA. Following DNA replication, the chromosomes condense in the nucleus of the cell. As a cell prepares to divide, chromatin coil up further, shortening and condensing the chromosome. The replicated chromosomes are called sister chromatids. First, duplication or replication of the chromosomes. Interphase is a phase of cell cycle that precedes a cell division. Conformed reproduction of chromosomes takes place during this phase. A homologous chromosomes each consists of one chromatid. During this period or during this phase, the chromosomal material doubles and leads to the formation of chromosomes having two chromatids, two sister chromatids that remain attached by the centromere. What is the role of interphase? A role of interphase is duplication of chromosomic materials. For example, take two chromosomes for simplicity, chromosome number 9 and chromosome number 23. Chromosome number 9 carries two alleles, A and B, and chromosome number 23 corresponds by X and Y. During interphase, the chromosomal material duplicated and each chromosome becomes two sister chromatids joined by centromere. Then, the cell undergoes mitosis, where the sister chromatids separates and each daughter cell carries two chromosomes from each pair. Therefore, these cells are identical to the mother cell or initial cell. Now, what we can conclude? We conclude that during mitosis, the number of chromosomes and the genetic information are conserved. First, start by interphase, which is a phase that takes place before each mitosis in order to duplicate or replicate the chromosomal material. Cell in this phase consists of cytoplasm, okay, enclosed with a cell membrane, nuclear membrane that envelop the nucleus, which contains nucleolus and DNA as a form of chromatin. Finally, the centrosome, which divides into asters and produce microtubules of a cell. The cell is now ready to go through mitosis, which consists of prophase, metaphase, anaphase, and telophase. During prophase, the chromatin condenses into chromosomes, and the chromosome becomes visible, appearing as two sister chromatids held together at the centromere. In animal cells, centroles play an important role in the distribution of the chromosomes in the dividing cell. The centrosome duplicates and transforms into asters. Mitotic spindle beginning to form, and the nuclear membrane starts to disappear. During metaphase, the chromosomes of two chromatids move to the center of the cell and line up along the equator, forming the equatorial plate. Anaphase begins by dividing the centromere that holds sister chromatids together. The two chromatids of each chromosome separate. The chromatids move toward opposite poles of the cell. This is polar ascension. Two identical sets of chromosomes, each of one chromatid, are obtained. At telophase, a cleavage furrow forms in the center of the cell. The chromosomes uncoil and go back to the state of chromatin. Decondensation of chromosomes takes place. The spindle fibers break down and disappear. The asters become chromosomes, and a new nuclear membrane forms. In animal cells, cytokinesis completes cell division by extending the cleavage furrow to completely separate the newly formed daughter cells. Since plant cell walls cannot be constricted by actin fibers, vesicles form an expanding membrane partition called the cell plate. Like animal cells, plant cells use cytokinesis to finish the division of the contents of the cytoplasm between the two identical daughter cells. Now, what is the difference between mitosis in a plant and mitosis in animal cell? Plant cell have no asters, while animal cell have asters. And plant cells, they divide by lamella wall instead of a cleavage during telophase of animal cells. This document shows the cells in different phases of mitosis. First picture is the cell during prophase, beginning of prophase, where the chromosomes are condensed and the nuclear membrane starts to disappear. While the second one is the end of prophase, since the asters at the opposite poles and the spindle fiber appear. Then the third one is correspond to metaphase, where the chromosomes of two chromatids move to the center of the cell and line up along the equator, forming the equatorial plate. Then anaphase, where the centromere divides and the two chromatids of each chromosome separate and move toward opposite poles. This is called polar ascension. Finally, telophase, where the cell constricted and the cleavage furrow at the middle of the cell.